Howdy guys, welcome to the show. So today we've got a lot of different things to talk about. So I first want to say hello to everyone who's here chatting with me. We've got Critical Cripple, Nana's here, Mr. Shadow Kitty. So what's up guys? Hey Exile, how are you? What's going on? So today we've got a lot of different things to talk about. I've got all kinds of stuff as far as, hello, hey. So I've got all kinds of different science discoveries that we're going to talk about. So I'm thinking I'm probably going to start moving towards talking about the latest in science research that's come out, because I've covered so many topics. And then anytime somebody has a topic they want me to talk about, then I'll put together something like that. What do you guys think? Because I'm thinking I probably need to, I really kind of want to start digging into current research and what that means for people, you know? So I think that's kind of important. Hello, everyone. So, it's been a pretty busy week. Been a pretty busy week, particularly for stuff in science. I've been digging around trying to find out some of the latest stuff that's come out, and there's all kinds of different publications that you can look at. So I've mentioned in the past, you can go to, let me pull this up, latest research news. Okay. No, that's not what I wanted. Here we go. So one of the places that I go to, let's see. I'll show you here. Hello, humans. Hello, everyone. So I'll show you here kind of one of the places that I go to. So here I got this, massive screen. So let me blow this up a bit. This is one of the places that I check out to get what's going on. And all of these are funded by the National Science Foundation, these particular research projects. The National Science Foundation funds a whole lot of research, and it's across the board. Hey, Bill Portley, just subscribed. Thank you. We get a little Stephen Hawking hopping around. What's up? So if you notice, I got things going on up here too. I've got the chat boss. So this is a boss up here. Can you defeat the boss? Knock out all my hit points. I don't know what happens after that. This is a new thing. So if you guys can defeat my boss here. And I don't know why this isn't scrolling. This is supposed to scroll. So anyway, this is also up at the top here. I've got super chats. Don't know why that's scrolling. That'll be a thing that I have to fix. But anywho, today we are learning about the latest and greatest in Hello Geek Room in science research. So some people have asked me about periodicals. Where can I go that's a good reputable type of website? I always kind of give them this website a good starting foundation for that. So it's nsf.gov/news. So if you just type in NSF research news, it'll take you to this site, and it breaks it down by the week. So brought to you by the NSF. I think this was probably supposed to say December, because it's usually in order by a week. So we're going to talk about a few things on here, but I'm not going to hit everything, but we are going to talk about a few things in here. Some really cool stuff. Geoscientists develop technology to improve forecasting of earthquakes and tsunamis. So what's up? So it's like, I wish I had a wrench so I could have given Kayla Swift the Swift boot. Yes. Let me give some people mods. I have a feeling it's probably going to be pretty icky, here and there, but that's all right. I can add mods right up in here. What's up, man? All right. So anywho, so we've got all of these going on here. So I'm going to touch on some of these. But another website that I really like, and I'm going to pull something out of Science News right here. I really wanted to talk a bit about this. What happens when governments crack down on scientists just doing their jobs? But that almost warrants just an entire episode, because there's so much information there. So this is a nonprofit journalism website. So Science News, do check that out. I am going to talk about a few of these things here. A Polar Affair. There's a book about penguin sex, guys. So if you didn't know, books come out about penguin sex. And there was, so this is not an NSF thing. I don't mean National Science Foundation. Not say NSFW, I guess. Or not safe NSFF, not safe for families. This completely safe for families. So don't worry about that. We talk about some things on this show sometimes that are just like, hey, Slim, what's up? So I think I'm gonna talk a teeny bit about that article. I'm not gonna spend too much time on it because it really does need its own show. Academics and scientists who are doing their jobs and basically have their basic rights kind of taken from them. just for doing their jobs in particularly strong-armed type of governments. So if it tells us about how penguins are getting more than us, it'll be depressing. It's not. It's actually, it's a good, let's just go ahead and talk about it. But before I do that, I want to kind of say hi to everybody and catch up on the chat. So this is the current setup. This is my current setup. It's kind of new. I like the animated background. Somebody is currently working on a new set of screens. And this one I think is kind of cool. But the other set of screens, I believe, are going to be 3D. So there's 3D screens. But this is how we've got it kind of currently set up. I like this. This is so cool. I haven't had like an update in a while. So let me catch up a bit on the chat. So hello, Slim. Yes, I have wrenched Mr. Shadow Kitty. Mr. Shadow Kitty almost sounds like a Watchmen character. I'm just saying. So yes. Hey, Math Pig. How you doing? So today I've got my pirate mug. I have it's full of coffee, but it's a pirate mug. Oh, before I start jumping too much into this. So tomorrow we have Dr. Amanda Glaze on the show for Oral Audacity. So it's pretty freaking exciting. She's been on my show a couple of times. I've done a podcast episode with her where we talked about the cultural resistance associated with evolution education. So tomorrow we'll talk a little bit about how she overcomes that and some of the myths that are associated with you can't learn evolution because it contradicts your belief systems. This is her bread and butter. This is her meat and potatoes. This is what she researches on how to overcome those obstacles to help educators in different parts of the world be able to, in within the U.S. even, we have a lot of resistance on evolution education. So Dr. Amanda Glaze will be on to talk about her research and some of the myths that are associated with I can't learn about evolution because it's a conflict of my personal beliefs. So she's going to kind of jump in on that. So it'll be kind of interesting. Oh, thank you, Exiled. Yo ho, a pirate's life for me. We pillage and plunder and don't give a hoot. Drink up, ye hearties, yo ho. So, that's probably my favorite ride at Disney World. One of them. I like that in Haunted Mansion, and they changed Space Mountain, and I hadn't ridden it since they changed. Hello, Amelia. So it's the pirate nose. Yeah, it's missing. It's a skull. Abandon all hope. No, that's not it. It's like the beatings will continue until morale improves. I have a pirate shirt that says that. It says the beatings will continue until morale improves. Y'all. It's funny. Having used to teach high school students, I probably should have hung that up. But that's really actually not funny. That's actually not funny not to have that, My teachers out there who have taught high schoolers, you often feel like that sometimes. But, of course, don't hit kids. That's not okay. We don't do that here. So, anywho, so I've caught up with you guys in the chat. Let's kind of talk about some of the things that we're gonna go merch that shirt. I should. The beatings will continue until science improves. That's probably for my graduate students out there. Those of you in grad school, the beatings will continue until science improves. That's going to be on a t-shirt now. For those of you who have been in grad school for science, y'all know what I'm talking about, man. You take a beating. Gosh. So, yeah, the beatings will continue until the science improves. All right, so let's have a look at some of the stuff here we got. So here, a polar affair. This one's pretty interesting. So I was like, I've got to see what's going on here. So essentially, All right, so this is a particular review. So this is a book that's come out, and it talks about essentially penguin biology associated with Antarctic exploration. So back in the day, there was a British explorer, Robert Falcon Scott. Now, if you haven't been to the London's Natural History Museum, part of their treasures exhibit has an emperor egg. And you can see that. So if you're in London, go to your freaking museum, man. You can actually see. And they did, one time I was there, they did a particular exhibit just on a lot of the pictures that they had associated with it. Now, he wrote in this final diary, essentially, about this quest to reach the South Pole. There was a naval surgeon and zoologist named George Murray Levick. He was kind of hunkered down at this snowbank, kind of observing the Adelaide penguins. Levick accompanied Robert Falcon Scott to Antarctica, but was not one of the 5 expedition members that went on to the pole. Now, this journey actually claimed the lives of everybody. But here's something that's interesting. This is where we get into the human element, where humans will suppress knowledge that conflicts their belief systems. All right? So during this time, during the months observing this, all right, they left out information. All right, so let me zoom in so you can kind of see this. Okay. So here, he left something out. During his months, now let me give credit here. This is an article by Carolyn Gramling. All right. So this is an article by Carolyn Gramling in Science News. During his months observing Adelaide penguins, which included an entire breeding cycle, Levick, he left this out of his book, witnessed the birds engaging in same-sex mating rituals. He also saw the birds engage in a variety of other sexual behaviors that in humans we might call promiscuity, infidelity, and even prostitution. Penguins are kind of busy. Levick recorded these scandalous details in a second manuscript called The Sexual Habits of the Adelaide Penguin in 1915, but the manuscript was stamped not for publication and remained unpublished for nearly a century. So, essentially, this was left out. And a lot of it, so you can actually find it. So you can find the manuscript. It's in a scientific journal. It was popped up in 2012. Lloyd Spencer Davis, who had thought he was the first to record same-sex behavior in 1996, found out, no, he did not. So, no, he hadn't. Levick had, but they hid it, because society. Suggested society. So this particular book, it's not really, this book, A Polar Affair, it's not really to try to understand why the penguins are doing this, but why would they discover the dirty side of penguins and then try to cover it up? So this book kind of dives into Levick's personal history, the field notes, but why cover up penguin sex? Why cover this up? So a little bit of societal roles, kind of playing a hand in hiding scientific information. Davis' discussions of whether there are evolutionary benefits to penguin same-sex mating or non-monogamous behaviors are fascinating. Is the same-sex mating a case of mistaken identity? That male and female penguins are monomorphic, looking much alike? Is promiscuity among penguins related to the female's inclination to build a stronger nest? So there's a lot of different questions that they kind of wrestle with. But you can pick up this book on Amazon if you want to kind of see as they're diving into Levick's background and that sort of thing. Why hide the penguin sex? What do you guys think? Why did they hide the penguin sex? I'm inclined to think that it's a little bit about, that wasn't allowed and they didn't need to have that reinforced. Hey, look, nature, this happens in nature, therefore it's natural, Prostitute penguins. Yes, that's right. Necrophilia. We see that with ducks. I don't know is necrophilia a thing in penguins? Let me look this up. Penguins and necrophilia. I bet if you guys looked at my search history, it would be pretty interesting. Let's see. Yeah. Okay. Yeah. All right. So yeah, you're right. It looks like rape, child abuse, and even necrophilia, it seems. Well then. And I thought ducks were weird. So they didn't want to admit the activity is monetized in nature. See? That's what I'm thinking too, Exiled. I think it's a society thing, but so yeah, lovable Madagascar penguins are known to rape and torture. So I learned something today. Let's see if we can destroy other things that we've learned. Here we go. How about this? Sea otters are jerks, so are dolphins, penguins, and other adorable animals. Thank you, Slate, for that title. So sea otters, they're not all that great. Dolphins are pretty jerky too. I'm going to ruin sea otters for you. So here, they're pretty. So check that out. Frolicking. Some sea otters have developed the disturbing habit of humping and drowning baby seals. So welcome to the show, guys. Saturday Morning Science. Yay! Thank you for the super chat, Angry Roach. What we got here? Dang, penguins don't mess around. No, they don't. Welcome to the show, guys. We're learning that I am ruining your animal loves. Thank you, Internet. They're ruining it for me too. I have found things out today. Oh, dolphins are rapey. I have actually swam with dolphins, and they were not rapey with me that one time. I do not discount that they might again. Dolphins get high on puffer fish. They do. Dolphins are extremely intelligent. I don't think people give them enough credit. And sea otters are just mean. Oh, yes. So we've learned some things today. Yeah. So look, you guys are knocking down my hit points. Don't forget. Also, I forget to mention this. Swag bag. December 15th is my cutoff day. That's the last day to earn Mel's Minions points for this quarter. Every 3 months, I think I'm going to throw together a swag bag of stuff. And I've got different things. Well, I had them over here. I have some stuff over there. I'll grab one of my, I have lots of different things in my background, guys. Doc Savage says I'm a hoarder of the geek stuff. That might be true. But I'm going to grab one of the things I have in my background, and I'm going to toss it in a swag bag along with a personalized limited edition postcard with a special message on the back. That's going to go in there. I also have a mug that I'm going to send you, old school mug, and a t-shirt. That'll be for the swag bag. So the person who has the most Mel's Minions points is going to get the bag. Now, I think what I'm going to do is every quarter, because some people are just really good at collecting them. So every quarter, somebody who hasn't won a swag bag yet will get a chance to get the swag bag. So, be certain you watch the show. We're every Saturday and Sunday. Watch the show and build up some points. You can play a little Streamlabs games on the side to kind of help you out with that, to boost your points. So you guys are pretty close. You're doing really well. So you can play along and boost up your points and get some free stuff. I already know what I'm tossing in this bag, and I've got stuff together for the next two. So it'll be awesome. All right. So let's talk next. How do we get points? You get points by watching the show. You can play along with the little Streamlabs games. There's, and if you want to do commands and see, I have different commands in YouTube. Now, this is for people who are watching on YouTube. The Streamlabs points don't translate to Facebook and Twitch and all that. So, unfortunately, I can't get all that there. But that's all right. We're working on it. So, let's see. And let's get back to what we were talking about with here. All right. So, that was A Polar Affair. Do check out that book. It's available on Amazon. And so you can kind of dig down and see a bit of the history about how society can influence whether or not certain stuff gets published. That is a thing. All right. How about, let's give this one a go. I found this one. This popped up pretty quickly. We've talked about in the past the evolution of dogs. Dogs were kind of... they evolved over time with our help. We selected for dogs that had the cuteness. My dog has one ear up and one ear that's floppy, and he's got a cute little face, and you're just like, oh, you just want to mush his face. So we have generated breeds of dogs for selecting for certain traits. We can now kind of think, okay, a gene tied to facial development hints that humans domesticated themselves. We domesticated ourselves. So we kind of went from the more protruding, forehead thing to a more, pronounced kind of what we would call like cuteness, less hostile looking, less aggressive looking, less hot-headed looking. So we've got like a picture here of modern humans on the left side and a little bit more of the Neanderthals on the right. So there's research to kind of implicate that there's a particular gene that affects those developmental features that kind of spread out and show in different areas. So we know that this has happened with dogs. We've seen it with horses. But the first incidents that we started to kind of go with that, this was around 2017. There was this idea that humans kind of domesticated themselves. So we started to select for certain features. But there wasn't a whole lot of biochemistry associated with those cells. There's something that's called, we've got something that's called, what is that cell called? Where is it? It's a neural crest cell. All right, so here's the idea. Neural crest cells, let me blow this up a bit. Woo, too big. All up in your face with the science. All right. So, in 2014, there were 3 scientists who kind of proposed that, we see this in dogs and horses and stuff. There's certain things that are for tameness, and there's slight genetic changes that we see in their faces. Their little aminal faces. They're so cute. These changes hamper movement of some developmentally important cells that stop it from progressing with that particular growth that we would see. Now, we see them early in embryonic development, but they kind of migrate to different parts of the body, and they become all of the other tissues that we see, kind of like what stem cells do. So these researchers, the idea was that mild genetic changes might produce neural crest cells that don't move as well, leading to these animals with this cuddlier look. Well, now we're applying that to people. So they had a study that came out, and they were looking at people with developmental disorders, to kind of learn what makes these particular cells tick. And so the gene that they found out, BAZ1B, that is the boss. That's the boss of these neural crest cells. And so what they found is that this gene controls 40% of the genes active in neural crest cells. So it's a huge boss. And it determines essentially how quickly they kind of move around in these different lab tests. Now, we don't know 1,000% or 100% that humans domesticated themselves, but this is kind of what we're starting to see. This is a little bit of some of that proof that humans kind of underwent self-domestication, which is kind of cool. But you can check out, let's see. They get into the detail of it. They just basically took a bunch of cells from people that had 2 different developmental disorders, and they got a big old piece of the DNA, and that's from what it says here is chromosome 7, that has the gene BAZ1B. And so the people that have Williams-Barron syndrome, they're missing a piece of DNA from number 7. And if you haven't gotten into like any time that you're dealing with a genetic disorder, usually it has to do with the presence of too many of a particular gene or a missing gene. So these particular individuals only had a copy of just one of those BAZ1B genes that regulate the neural crest cells. Those people tend to be characteristically talkative, outgoing, not aggressive. They also tend to have these little round faces with short noses and full cheeks and wide mouths and full lips. So they're looking at the presence of certain behavior characteristics in addition to what we see manifest through them. And so while we know that dogs that are more mild-mannered and cuddly and cute are affected when we select for those, that cuteness and that cuddliness, we haven't been able to definitively show that that was associated with their tameness. So this has kind of given us more information that the less aggressive, outgoing, friendly type of characteristics may very well be associated with BAZ1B. Isn't that interesting? So you can find this on sciencenews.org. This particular article is written by Tina Hesman-Say. If I pronounced that wrong, I'm very sorry, Tina. And that's just come out like 3 days ago. And again, this is a website that's completely free, and you can kind of check out the latest information on that. Let me catch up on the chat. What's up, guys? Alrighty. We be hanging out and stuff. This is all pretty interesting. Hello, frustrated atheist. Tony Design showed up. I'm just going to sit here and eat my popcorn and watch you and Exiled just go at it. Central is the best time. Oh, my goodness. All right, so we got a lot of people going. What time zone is Mel? Central. I am Central Standard Time. I am Central Standard Time, so that's awesome. And tomorrow at noon Central Standard Time, that's GMT -6. So if it's 6 p.m. in the UK, it's noon here. Greenwich Mean Time. That Greenwich time is so mean. They're not very nice, are they? Anywho. Tomorrow, noon Central, or Audacity with Doc Savage. So do check that out. It's about the heist. Okay, so I'm checking out the chat here, trying to catch up. Don't make me time you out, Math Big. Oh my goodness, you guys are silly. Alrighty. Tony Design, 72,694 Mel's Minions points. Dinah Cat loves me. You guys are playing along. I'm getting points. I don't know how I'm getting points. I guess because I'm here. I get points for showing up. But there you go. Alright. Catching up. We were just saying how Tony is winning. We were. You came in late on the day you're doing genetics. Well, it's a little bit of genes. Am I free on Tuesday at Mel 4 p.m. your time? 4 Central. I will have to look that up. Let me see. This Tuesday, I have a meeting at 6:30 Central. It's for a local group that I work with for pop-up science events in the area, and we're planning our science festival, our spring science festival. That's at 6:30. I could possibly do a stream, but it will have to be from my phone, maybe at my job. So, because that's till 2, and I have a thing at 6:30. Probably not the best day. Maybe another time, Math Pig. If you were asking me to do a thing, that might be, another time. All right, so ScientistMel, give me all the points. You have to play along with the little Streamlabs games. Alrighty, so that kind of helps. Whoops! Oh my goodness, you guys are funny. All right, so we might have domesticated ourselves, guys. Are we not surprised? I'm not surprised. I'm not surprised at all. All right, so let's have a look at something else. So I had mentioned this website, right? There was all kinds of stuff here. We're done talking about penguins and being jerks and stuff. All right, so again, another National Science Foundation. Good site to get information on the latest in science research. Completely free. And oftentimes, if you want to click on the article, there's usually not a paywall. And so you can go through and actually read the science that they're doing. So I like that, because a lot of these, so if you're like, I want to be able to read some science papers, but paywalls are a thing, ugh. You're like, hey, fear not. Go to National Science Foundation, and most of the time, it's a rare day that I come across that. Most of the time, you can click on the news articles, and they'll take you, let's see, like right here, how mantis shrimp makes sense of the world, and what they do. Usually you can click on it and they'll have a full text available. Let's see, is this a paywall? Blast it. Okay, so that one's a paywall. So thanks a lot. But this one, for example, sea fan corals face new threat in warming ocean, copper. So if you're like, I'm worried about the coral and copper stuff, 133. Oh no, Nana attacks me. 666 through Super Chat. Thank you, Nana. How you doing? Streamlabs is rigged. You do get points for doing Super Chats. I think you get, I don't know how many points you get, but you do get points for doing Super Chats. So, they can be, so yeah, hello, Robert. You didn't hear it from me, but I think Tony Designs has a system of giving himself points, but don't tell him that. I'm on to him saving it as a surprise. But the thing I like about what is going with the National Science Foundation, essentially, okay. Did I freeze for a second? Am I back? I'm back. It looked like I got all weird for a second on OBS. Anywho, as I was saying, I really like National Science Foundation because of the fact that most of the time you can gain access to the articles without any kind of worrying about paywalls. And so here's one. So let's talk about sea fan corals. Warming ocean. Copper is an issue. And this came out about, let's see, where is it? December 2nd, 2009. So about 5 days ago, this information came out. So let's have a look. So essentially what we're finding is sea fans. And so these neat little sea fan corals, they're highly susceptible to copper pollution. So this is a big problem. And what it does, it affects their immune systems to where they are more easy. It's easier for them to become diseased. So that's a big problem for them. And this particular paper was published in, let's see, let me blow this up. Ecological Applications, ESA. So it's the Ecological Society of America. So warming in coordination with copper metal being in the ocean affects the immunity of these different coral sea fans, and it kind of ends up causing them to be more susceptible to diseases. So let's blow up a bit more. Here we go. So hopefully you can see that. So warming is a big issue, but also acidity is a big issue when it comes to coral reefs. We don't think about that. An increased amount of carbon dioxide in the water, that's the bubbly in our water, increases the acidity in the ocean. So what they did is they investigated how warming oceans and copper pollution affect host immunity and disease risk for 2 diseases of the abundant Caribbean octocoral, known as the sea fan Gorgonia ventalina. And so they found that cellular immunity of sea fans increased by 12.6% at, field surveys across the sediment copper concentration gradient in Puerto Rico. USA revealed that cellular immunity of sea fans increased by 12.6% at higher sediment copper concentrations, while recovery from the multifocal purple spots disease. Multifocal purple spots. Say that 5 times as fast as you can. Multifocal purple spots, multifocal purple spots, multifocal purple spots, multifocal purple spots, multifocal purple spots. I found a new mouth thing for actors and singers now. Say that, multifocal purple spots. I'm sorry. I used to be in theater, and that was the thing we did. Aluminum, linoleum, aluminum, linoleum, that sort of thing. So yeah, anyway. So words are hard, So what they found was while recovery from multifocal purple spots, MFPS, tended to decrease. So they had a harder time recovering from the disease when they were in a higher copper environment where there was a lot of copper sediment. And so tissue damage was also greater in treatments with higher copper and warmer tissues. So you saw more tissue damage in these coral fans. And so what they think is lab evidence confirms that elevated copper hinders the sea fan's immune defenses also against damaging parasites. So there's all kinds of just awful stuff that's going on with these poor corals. So essentially what they're saying is this is the first evidence that metal pollution affects, this is where they say right here, this is the first evidence that metal pollution affects processes influencing disease in octocorals and highlights the importance of immune mechanisms in environmentally mediated disease outbreaks. So essentially, the more metal pollutants you have with this particular type of species of coral, the less likely they are to recover quickly from standard diseases that they're subjected to. So, and you can check out this research. This was done by at Scientific Cornell Institute, Allison Tracy, Ernesto Weil, and C. Drew Harvell. Give them a shout out and have a look. I believe this was Cornell Institute. Yep, Cornell University. All right. So do check that out. We still got some time to talk about some things. Let me catch up on the chat. You guys are playing around and stuff, learning. Learning is like knowledge is power, right? How now, brown cow? So that's right. Let's see. Multifocal purple spots. I saw them in concert in 1989. That does sound like an 80s band, doesn't it? It does. Mercury is a big problem. You're right, Robert, it is. Let's see. Definitive proof. Are we going to do a scientific study on Tony Design's means of hacking the point system? Are we going to spend 4 years hollering about that? 4 years of that. Stuff and things, my faith. Oh my goodness, you guys are funny. The more you know. So, let's see. There are some other things I wanted to talk about. Let me pull this. All right, so I'm going to ask you guys what you think. So I've mentioned that I do, and if you just jumped in a bit ago, I mentioned that I'm probably going to spend an entire episode just talking about academics and scholars, as well as scientists who are persecuted by governments just for doing their job. But there's a lot of information here, and so this came out yesterday. Let me find, where is it? It's right here. So if you want to check this out, do go to Science News, and the title is "What Happens When Governments Crack Down on Scientists Just Doing Their Jobs." Well, there is an organization that's come out just to try to help these individuals who are in more oppressive type of regimes in different countries that have a strong arm on science and knowledge, and they don't want certain information to go. And it actually gets pretty grim. There is one scientist that was accused of being a spy who's now been sentenced to death. Other scientists that have been charged with promoting propaganda by asking for investigations associated with the mistreatment of certain people. It gets pretty scary. Medical professionals who are shot and killed. So I think I probably need to dedicate an entire episode just to that, because it's pretty scary. But human rights take a back seat when state leaders try to control the narrative. So if you want to kind of dive in and have a look, this is a pretty good piece by Sujata Gupta. I'm not certain if I pronounced that correctly, and if I mispronounced that, I'm very sorry, Miss Gupta. Gupta, could be Gupta too. So I want to apologize if I mispronounce your name. That's on me. But it's a pretty thorough piece, and it goes through a lot. Here's an example of Madrasa Jalali, I think, was in Tehran, and she was trying to do a training program for Iranian hospitals. She was, sentenced to death. He was sentenced to death for being a part of that, just trying to do his job. So I mean, it's a pretty grim read. So scrutiny of Chinese scientists, charges against Chinese named people, espionage charges, scientists in Turkey. I mean, it spans the globe. But if you want to kind of see how you can help these people, let me throw this out there. There's a website called scholarsAtRisk.org, and they work to help these individuals who are persecuted for what they do, their job. So Free to Think 2019 is the 5th installment of an annual report by SAR's Academic Freedom Monitoring Project, analyzing 324 attacks on higher education communities in 56 countries between September 1st, 2018 and August 31st, 2019. So give that a look and see if you can help them out. You can get the report here and kind of see what's going on. ScholarsAtRisk.org, protecting scholars and the freedom to think, question, and share ideas. So kind of head that way and maybe raise a little bit of awareness. So I will probably spend an entire episode just talking about those sorts of things. So it's pretty scary stuff. So let me catch up on the chat. Yeah, it is pretty horrible. That organization, you can get involved by joining the network. And if you're in academia, you can connect your campus with one of the below opportunities. So, guys, if you're in academia and you're watching this, here's how you can help. Let's see. So there's opportunities. You can join the network. You can host a scholar. Member institutions invite threatened scholars to temporarily join their campus communities as professors, researchers, lecturers, visiting scholars, postdocs, graduate fellows, students, anybody. So you can host somebody at your college. You can give them a place to speak and talk about their experiences to kind of raise awareness. There's also seminars, and there's also researchers that partner the Academic Freedom Monitoring Project. So they go through and they collect all the information that's associated with academics and scholars who are at risk. And there's you can work in groups, you can do workshops and conferences. So this is kind of geared more towards academic institutions and other academics who kind of want to help raise awareness for this. But you can donate. They have a Twitter. You can subscribe. And just, just raising awareness is exceedingly important. And if you need help, if you're a scholar, an academic scholar or an academic or a scholar or a scientist who's in a problematic area, you can do that. All of these things, how to join the network, their mission statement, their publications, scholar resources, all of these things are available to you to kind of help raise awareness and help those individuals who are out on the front lines getting all kinds of, having their human rights violated. So there you go. Goodness, so I'm catching up. People are so angry. Oh my goodness, fear not. I will give away a swag bag every quarter, so every 3 months. And if you've already won a swag bag, if you win this one, I'm gonna like have you have a timeout for like the next two, so somebody else can have a chance to win. You know what I mean? Let's be fair. Have them. So if you don't win the swag bag this time, fear not. We're going to give you an opportunity. If you've already won one, you're going to have to sit out the next couple. Maybe share your points gaining knowledge with others. You could do that. I'm looking at you, Tony Designs. There you go. But hey, it's all right. I want everybody to have an opportunity to, and to get involved. So it'll be cool. It'll be all right. You're going to be okay, Tony. Secure the bag. All right. So let's cover one more topic. What do you guys say? Let's cover one more topic. Let me ask you what you would like to learn about. You want to learn about the mantis shrimp? Do you want to learn about technology on improving the forecast of earthquakes and tsunamis? Do you want to know more about Alzheimer's? What do you guys think? What you think? Which one would you like to talk about? Earthquakes and tsunamis, Alzheimer's, or the mantis shrimp? What do you think? Nobody knows. You guys are playing for points right now. All right. I'm just gonna pick. Somebody needs to pop up and tell me, man. Pop up and tell me shrimp. Shrimp was the first one said. So let's talk about the mantis shrimp. And we can do Alzheimer's another time and talk about, and I can carry over. We can talk about the geology stuff at a later date. So let's talk about the mantis shrimp. How cool is that? Alzheimer's. If we have time, we will do Alzheimer's. I had to turn off my heater. I have a heater down here and my legs are on fire now. Alrighty, so here we go. So this comes from the National Science Foundation. How mantis shrimp make sense of the world. How do they process information around them? Are they stimulated and that sort of thing? So essentially, they do use different visual information and other sensory input in order to help them understand the world around them. So this was popped over into Journal of Comparative Neurology. And so these teeny tiny little small brains of these mantis shrimp, they are very, very, very fierce predators, and they have really awesome vision. But they kind of like process and integrate this information with other sensory input through their brains. So out of the University of Arizona, they got together, the research there, Nicholas Strassfeld, got with the other ones, and they kind of wanted to understand better how their bodies connect to other parts of the shrimp brain. I like shrimp, and I'm really hungry right now. I had some gumbo this week. I was not impressed, and I'm kind of sad about it, but I'm not going to talk about that. So, mantis shrimp, they've got this like stereoscopic vision. So they have a huge band of what we call photoreceptors. Photoreceptors are receptors that can process photons, different types of light on different wavelengths. Now, if you're not familiar with how it is that we see color, we have different photoreceptors that go within what we call the visible spectrum. Right? And so light travels at specific types of wavelengths, and the wavelength that they have is the color that we see. So I believe, I'm trying to think, I think it's around 700 nanometers of greenness. So let me pull that up. Let's just do this. Visible spectrum wavelengths. So let's just pop this up. All right. It's not 700. 700 is red. See, I'm wrong. All right, so here. So the distance between waves of photons, that's our wavelength, and so they're traveling, the size of the waves, right? They're traveling, and we usually see this about nanometers. So the ones that are about 550 nanometers apart, that's green. All right, so how the big the waves are kind of dictates. Here's probably a better one. Here we're into infrared. So 700 is pretty huge. Once we get past 700, we get really big wavelengths. That's where we stop seeing colors, because our photoreceptors, our eyeballs, aren't able to process wave photons that are moving at that particular wavelength, and frequency, right? So these particular researchers, what they wanted to do, let me pull them back up here. So the mantis shrimp can distinguish up to 12 different wavelengths and linear and circular polarized light. And so it seems like they're able to process like all of these different channels of information using their reniform body. Okay. So these reniform bodies have like these little centers that are called mushroom bodies. All right. And this is kind of like what we see with arthropod brains, and they're required for olfactory learning and memory. So for us, our olfactory is like really close to our nose. That's why we associate like a lot of memories with scent. That's right where our memory centers are in people. So what they said, and this comes straight from the fact that we are now able to demonstrate that the reniform body is also connected to the mushroom body and provides information to it that suggests that olfactory processing may take place in the context of already established visual memories. And so they have this processing that's established with the mantis shrimp's memory. So they remember things. I wonder if, if mantis shrimp can remember things, do they hold grudges? Can mantis shrimp hold grudges? That's what I think the next question should be. What do you think? So if you want to take a look at that paper, the reniform body, an integrative lateral protocerebral neuropil complex of Humalis castraca identified in stomatopoda and Brachyura. I said all of those words. And this was first published in October 2019, I guess. It was maybe the preliminary publication before they did any kind of revision. But you can check that out. And how do they get this information? Using confocal microscopy and classical silver staining, we demonstrate that the reniform body receives inputs from multiple sources, including the optic lobe's lobula. So optic lobe. So optics has to do with eyesight. So that particular lobe associated with visual processing is connected as well. So that's pretty cool. Although the mushroom body also receives projections from the lobula, it is entirely distinct from the reniform body. So the fact that the two are separate, that's really big, huge news for shrimp biologists. You wouldn't know that, and I wouldn't know that either because I'm not a shrimp biologist, but this is big stuff here. So that's essentially what they're saying. So we discuss the implications of their coexistence in Stomatopoda, the occurrence of the reniform body and other Eumolluscostracan lineage, and what this may mean for our understanding of the brain functionality in Pancrustacea. That was a lot of words that I'm not used to saying, but that's okay. Chemist. It's all right. We're good. We're chilling. We're saying interesting words. So that kind of gives us a little bit of information about that. Hi, microbloganism. How are you? Hello everyone. So yeah, we're just kind of catching up. Shrimpologists, yeah. Shrimpologists, so biologists that delve into the world of the shrimp, and this one happened to be on the mantis shrimp. I am King Frustrated, expert in bird law. There you go, frustrated atheist. Oh my goodness, I think my Streamlabs, did my Streamlabs freeze? It might have. I will have to refresh it. My Streamlabs restream chat. I'll just pull it up again. It'll refresh. All will be well with the world once more. All right. So, guys, we've kind of reached at an hour. So kind of to just wrap up a bit. Information, where did I get my things? I went to sciencenews.org and got the latest information. This is a nonprofit website, so you can have a look and look at the latest stuff. We talked about a polar affair. Penguins, they do some freaky stuff. And there's a new book out that talks about one of the first researchers of penguins who discovered a lot of information and how that was suppressed. You know, we can't be letting people know that penguins do some interesting things in their sex lives because humans are easily offended by everything that doesn't conform with their particular belief systems. Speaking of which, tomorrow at noon, Doc Savage and I welcome Dr. Amanda Glaze and her research that she does on evolution education for those who are resistant to learning evolution and how you can learn about evolution and it not conflict with your belief system. And so she's going to hop on here and talk about what she does with evolution education. Last week we had Tony Reed on, so that was pretty interesting, leading up to how can we resolve this? How can we teach evolution to individuals who are culturally resistant to the process? So you can learn about things if it conflicts with your belief system. It's absolutely fine. And Dr. Amanda Glaze will be on tomorrow at noon Central for Oral Audacity, and we will debunk a lot of the myths associated with I can't learn this thing because it's against my belief system. So we're going to tackle a lot of that. So do check that out. We've talked about gene tied to facial development and hence humans domesticated themselves. How is this a thing? Well, we domesticated dogs for cuteness. Well, we're doing that for non-aggressive people. So it seems like, but that kind of makes sense. Those individuals who are less aggressive, we now have genes, and we got to it's very preliminary. We need more information. We need more research to kind of definitively make that tie in. But there's more evidence to support the idea that humans domesticated themselves and selected for less aggressive individuals through the way that they look and behavior. And there's a gene for that they're finding out. Still need more information though. So we did that. What else did we talk about? We hadn't got to the Alzheimer stuff. We did talk about a polar affair. Then we hopped over to the National Science Foundation and took a look at some things. We talked about the mantis shrimp. We've also talked about sea corals, sea fan corals, and the increased amount of copper and how that affects their immune systems and makes it harder for them to recover from disease. We've talked about a lot of things. We'll be talking, I'll probably pull in Alzheimer's, and we can talk more about geoscientists and all of their tech that they associate with earthquakes and tsunamis. So if there's stuff that you want to look at and you hadn't had a chance to check it out. Science News, that's one website that has really good information, and also research news from the National Science Foundation. How awesome is that? This has been a cool talk. So let me check up on the chat and then we'll sign off here in a bit. Flippers are resistant to learning anything above a 3-year-old level. Oh my goodness. Let's see. Culturally resistant is the nicest way I've ever heard it put. I've written articles associated on how to teach evolution to those who are resistant to it. So that's kind of because I've had to do that before. Being in a classroom with a bunch of farm kids in the Bible Belt where they're resistant to evolution. And so how do you overcome that? How do you overcome that and make certain that they understand what the terminology is, what the words mean, without causing offense? So it's a thing, and there's research for it. Dr. Amanda Glaze. Speaking of non-conforming things, Doc Savage. Yes, Doc Savage. I am so lucky I get to call him my friend. He's been really busy this week, but he looks forward to spending Sundays with me chatting about the different things. And I adore Doc. He does a show with Omni Eris, and Omni Eris will be back on the 15th. So this Sunday we've got Amanda Glaze. Tomorrow, the following week, Omni Eris is back. And then in January, we're going to take a couple weeks off for the holidays. And then in January, I have Science First on, who does science journalism. And we'll talk about the misrepresentation of science in journalism. So that'll be pretty interesting. I think it'll be a good talk for Science First. So they'll be on to kind of talk about that. And if you have suggestions for guests for Oral Audacity, please do let me know who you would like to have on the show, and reach out to them, or at least reach out to me and tell me how I can contact them, and I will do that. It's nice going through science news with me. Yay! I like science news, and I can explain why things are important. Oh my goodness. Feral humans. Have ScientistMel cover that topic? Feral humans. Yes. We could talk about feral humans. I think they have access to the internet. I'm just saying. I think they have access to it. They're very aggressive individuals. I wonder what their bone structure looks like on their faces. That would be pretty interesting. Hey, scientists out there who are looking at, domesticating humans, get online internet trolls, check out what their facial structures are, and then coordinate that with behavior. Wouldn't that be something? That would be something to do. That might be a thing. Looking at the facial structures of the more aggressive individuals, that'd be kind of neat. Oh, goodness. Yes. So guys, you know what? This is original artwork made just for the show, by the way. And this piece is called The Visitors. The Visitors Associated, and by Livid Unum on Twitter. So you can check out his stuff. So we have original artwork. And so also you can go on my website, ScientistMel.com. I have merch there, and different stuff and things, mugs and all kinds of stuff, and artwork if you like that. It's pretty cool. Yep. A meta study coordinating internet troll facial structure with antisocial behavior. It might be a thing. They've already associated troll behavior with having the dark tetrad, So, psychopathy, I think it's sadism, Machiavellianism. It's a couple other things. Or I think it's the triad or the tetrad. But I talked about that in the science of trolls. And I've also talked about that in online narcissist behavior, So do check out my website. I have articles there. I post slides from my shows, from my informative shows. Like, share, and subscribe. I guess I'm going to read from the good book of Mr. Rogers. And Miss Mr. Rogers. He was an amazing human, and I don't know how he could... I don't know how he could get through the current world the way it is. Gosh, can you imagine Mr. Rogers being around right now trying to do what he does? If he started out now as a young person and trying to do what he did, it's just wow. All right, so the world according to Mr. Rogers. You rarely have time for everything you want in this life, so you need to make choices. And hopefully your choices can come from a deep sense of who you are. So... important lesson on time here. Be careful. And essentially, your time is valuable and your time is important, and how you choose and what you choose to do with your time and who you choose to spend your time with, that's a gift you can never get back. So you rarely have time for everything that you want in this life. So you need to make choices. And hopefully your choices can come from a deep sense of who you are. So what do your choices say about you? How do you spend your time? And is your time valued in who you spend your time with? That's particularly important too. Think about it. Y'all got to make choices. And then if you want to quote Gandalf, all we have to do is determine what to do with the time that we're given, right? Along those lines. They're probably not perfect, but there you go. It's a beautiful day on my Discord server, a beautiful day. Would you be mine? Could you be mine? I have always wanted to have a neighbor just like you. I've always wanted to live in a digital neighborhood with you. So let's make the most of this beautiful day. Since we're together, we might as well say, would you be mine? Could you be mine? Won't you be my science neighbor, my digital neighbor, my online neighbor? Just kindness is free, Kindness is free. Sometimes we make bad choices. Go watch A Beautiful Day in the Neighborhood. It's about an interviewer who learned how to control and confront his emotions because of our late and great Mr. Rogers. I think Mr. Rogers had such a high level of emotional intelligence. There's different types of intelligences, and I probably should do a whole episode just on that. Emotional intelligence is extremely important, and I often find that tends to go by the wayside, and we kind of see a lack of that, particularly with things that happen on the internet. It's a lack of empathy and consideration and understanding another person's position. That's kind of difficult, but hey, we do the best we can. So here we go. I'm going to go ahead and sign off because I've got things I got to do. I got to get ready for the show tomorrow. We have a new producer. I think it's Tronic Monkey Pants. Runs Hannibal and Monty's show. Going to be running Audacity for me. So I'm excited. We had a few bugs last episode, but there's always bugs on my show. It's not a ScientistMel show if I don't have some kind of hiccup, So there we go. Well, I'm proud of you guys, and thank you for hanging out here with me and doing science with me. So I'm going to say, you always make each day special for me. You know how? By just your being you. Check us out tomorrow on Audacity. That's the cheeky show where we swear a little bit, because Doc's savage. But Saturday shows are always family friendly. The Sunday shows, kind of like family friendly, meaning like teen and older. Sunday shows, definitely PG-13 and more higher, because, it's our cheeky, fun, adult, funny show. So I will see you guys later. So I'm sending squishy science hugs. Talk to you guys later. Bye.